So welcome everybody. Can you hear me, by the way?
Yes, I can hear you. And before I start off, I want to also introduce my colleague from 10x Genomics, Jose Jacob, who is present among the audience. And he is the product manager for the upcoming products that I'm going to talk about mostly. And so, yes, he's here to help me answer any questions that you might have about the product itself and about the science that it can help us pursue.
Okay. Thank you very much. So indeed, thank you for introducing Jose Jacob. And Nur-Taz Rahman from 10x will talk about their new product, Beam, right? Uh,
Yes.
and how it is helping system immunology study of the COVID. Just a reminder of where we are. So that's the iReceptor Plus seminar series, and we have two talks. We give like 45 minutes or a little bit more or less for the first talk, and then questions, which it's up to the speaker to decide whether you want to accept questions during the talk or not. You can say it. And then if we have some time, we'll take a little break and come back at the round hour for our second talk, who will be by Kazuyoshi Ishigaki from Japan. And that's it. So thank you very much, Nur-Taz, and let's hear you.
All right. Thank you so much, Professor Yari. I'm going to share my screen, and during my talk, it is okay to stop me for any questions. I might not be able to see my Zoom screen, so Professor Yari, if you can help me coordinate any questions if hands are raised, then that would be great. Thank you.
Okay, I will. Thank you.
All right. So I am going to see. Sharing my screen here and starting the slideshow. I really want to thank the organizers of iReceptor seminar series to give me the opportunity to speak as a keynote speaker. It's a real honor. And today I am bringing you some information about a new and upcoming product called Barcode-Enabled Antigen Mapping, BEAM for short, and I will refer to it as BEAM for the rest of my talk. And using this technology about 2 years ago in 2020, when we were in the first throes of the COVID-19 pandemic, scientists at 10x Genomics got together to look at the PBMC, peripheral blood monocytes of COVID convalescent patients, so somebody who had just gotten COVID and recovered. And we used the concept that we wanted for BEAM technology, and we looked at their entire adaptive immune system and their PBMC environment. So that is the data that I'm going to present to you, which is more of a proof of concept for the technology kind of data. We see some interesting trends in the data that I'm going to present to you, and we can talk about the exciting science that this technology will soon enable you to answer. Myself, I am, my name is Nur-Taz Rahman. I work with the Applied Bioinformatics team at 10x. If you write to software at 10xgenomics.com with some software issues you have, then it might come to me and I might help you solve your software woes. This is the iReceptor seminar series. So with this slide, I am not trying to educate anyone on what is the structure of BCRs and TCRs. But the point of having this slide is to illustrate that with the current product available from 10x Genomics, the single cell immune profiling product, we are already able to inquire for a particular sample type, what are the VDJ receptors, and get full-length sequences for VDJ chains for TCRs and for alpha and beta TCRs and IgH, IgK, IgL chains. When we look at the existing 10x Genomics 5' immune profiling product, the way it works is that we have the cells which we process in gem wells of the Chromium chip. And from the single well, we can produce gene expression library and VDJ library and sequence them with compatible sequencers, and using 10x Genomics's software, the Cell Ranger software, we can convert these BCL files into FASTQ files and then process them with the Cell Ranger multi-pipeline to produce our feature barcode matrix that we can use downstream. There are the VLOOP file and CLOOP files, which we can use in Loop browser software from 10x Genomics to do integrated visualization and analysis. And what Cell Ranger also does once it processes the FASTQ files, it's doing cell calls, getting rid of background gems, and assembling VDJ sequences, etc. So with this platform, we can do high resolution gene expression and immune profiling data analysis. We can find full-length sequences of the VDJ receptors, and we can also carry out clonotype grouping. What this technology that's currently present does not allow us to do easily and cheaply is it doesn't allow us to ask what antigen is my receptor binding to. I see an expanded clonotype in this sample. Why do I have that? What is it recognizing? Those things are currently difficult and might be expensive to generate in the lab, and the data analysis for those might also be difficult. So with the BEAM product, we are producing an end-to-end solution so that you can ask this question and generate data for it easily and cheaply. And at the same time, you have the software portion that is analyzing this data for you, showing you what the antigen specificity scores are. It is showing you what your clonotype grouping is and overlaying this antigen specificity score on the clonotype grouping so that you have no bottleneck in analyzing the data. You don't have to worry about building massive custom scripts of your own in order to do this. I'm an applied bioinformatician, so I had to emphasize on that software part a little bit, but a huge amount of work has also been done on the workflow side to make this easily achievable. So let me talk a little bit about how the BEAM technology will, apart from doing everything it's doing now, also allow us to screen the specificity of antigens to TCRs and BCRs. So moving on to the overview of the BEAM technology. The BEAM product is going to come in two flavors. So on the left-hand side, you see this schematic is labeled as BEAM AB, and the right-hand side schematic is labeled as BEAM T. So BEAM AB is where we are inquiring, is the product that is inquiring what are the antigens, which of my antigens of interest, target antigens, are binding to the BCRs in this sample. BEAM T is asking, what are my peptides of interest that are binding to the TCRs in this sample? What you can see is in the BEAM AB schematic to the BCR, there is a blue big antigen that is binding, and to that you see there is a complex that is added with the gray, red, and the purple string. So, and similarly, you see the same complex that is tied to the green MHC that is depicted here in this cartoon. So let me tell you about what those are. Those are the 10x supplied streptavidin, PE fluorophore, and feature barcode complex that we will provide to you. So for the BMAP product, what you are going to get from 10x is this streptavidin, PE fluorophore, and unique feature barcode complex that, the only thing that our users will need to bring for their experiment is their target antigen that has been biotinylated. And the way that they will do this biotinylation, they can do the QC, we provide guidance, complete guidance for that. Now for the BT product, we have the same complex, but we are providing it already conjugated to a MHC tetramer complex. That way, you don't have to worry about where you're going to get your tetramer from. You don't have to spend money getting expensive tetramers from somewhere. It's already provided. And it comes currently, we are providing 4 different human alleles and 1 mouse allele for it. And with that, you just bring in your own peptide that's of interest to you, you conjugate that, and you are good to go. But you might ask, so yes, Nur, I have conjugated my antigen or my peptide to the MHC complex, now what? Then the workflow is really simple. You have labeled your cells, probably unsorted PBMCs, with this conjugate for either BMAB or BMT as appropriate. And then once the staining is done, you do FAC sorting for that PE fluorophore. So you are getting on that PE color on your fax machine. You're FAC sorting B cells or T cells respectively. And then you move on to gem generation as per usual, sequencing, and you go on to data analysis. So it's a very easy workflow. Now, this workflow that I just described has come through a product development process 2 years ago, even before I joined 10x. When this data was generated, scientists got together and came up with the process. So I want to explain to you the experimental design for the work that was done, and for which I'm going to show you some data. For this experiment, the sample, as I have mentioned before, were PBMCs collected from a human survivor of COVID-19 and who had recovered at the point of when their PBMCs were collected. For the BMAB experiment, the B cells were enriched using an enrichment kit. And for the BMAB experiment, we inquired the BCRs with 2 different COVID antigens. One was the COVID spike antigen S, which in one of my figures is labeled as WT. And that just means that this spike antigen was the first, was from the original Wuhan strain that came about. And we also have the D614G mutation, which arose in March 2020 and then quickly became the predominant mutation by April 2020. And so these are the 2 antigens with which we inquired these donors' BCRs. And we also trimerized human serum albumin HSA antigen because we wanted to use that as a negative control. And these were complexed with total 60 streptavidin reagents. And each kind of antigen was then conjugated with a unique barcode so that we could then, we could get a readout for that, as well as there was a fluorophore. For BMT, the PHC tetramers were constructed by loading the pathogen-derived peptides, and there were about 9 target antigens here, 9 target peptides, and I'm going to show you what those were and what sequences they were at a later slide. And these were biotinylated into MHC monomers that from the A0201 allele. These were then tetramerized using streptavidin-associated fluorophore, kind of like the one that I showed you in our product right now. And it also had the unique barcode. So for BMAB and BMT for this experiment, so we got the PBMCs for the patient and we from the donor, and we carried out BMAB and BMT experiment. And so from the same gem well, we generated gene expression, VDJ, as well as the BEAM library for both BMT and BMAB, and we sequenced them and we did the data analysis. So after doing these 2 experiments on portions of the cell, about 8K and 5K cells respectively, the additional 100K cells were taken, and we just carried out gene expression and immune profiling analysis on those using our current immune profiling product. So no BMAB or BMT experiments were done on these 100K cells. For analysis, we mostly use 10x's software, Cell Ranger software, and then the Loop browsers that I have mentioned before. So via Cell Ranger, we achieved cell calling, we achieved clonotype grouping, VDJ config assembly, etc. For when this work was being done, we generated some plots and graphs for clonotype grouping, and those at that point in time were done with a freestanding software called nClone. That is a tool for clonal grouping of cells. Some of you might be very familiar with it. It's very easy to run on the command line. But currently, as soon as Beam launches, we are also hoping to release a newer version of the Loop browser software that is going to make the clonotype distribution plot as well from the clonotype grouping data. And for this work that I am presenting today, some custom scripts were also used to do the analysis at that point in time. So I want to move on to presenting to you the data that we gathered from the BMT experiment. This is the list of the peptides that we used to inquire the TCRs of this donor. What you can see are we have three peptides for the SARS-CoV virus. One is the spike protein. This is again one of the earlier spike proteins that were there. Then we have the ORF1, ORF3 sequences. We have EBV sequences as well as flu peptides. We also have an HIV GAG tetramer, which acted as a negative control, negative target antigen. So this heat map here shows us what we found from the T cell from the BMT experiment. Let me take you through this heat map. So once we carried out our gene expression VDJ and Beam analysis, what we got from the VDJ part was the clonotypes. We received what were each sequence of each of the TRA, TRB chains, and then how they were grouped into clonotypes. So that clonotype is represented in this, those clonotypes are represented in this heat map. Each row of this heat map is a clonotype. And on the column, we see the peptides. Antigen specificity for each of these peptides were calculated by comparing the UMI counts for the target antigen against the UMI counts for the control antigen, which in this case was the HIV GAG. So what we are seeing on this heat map is binarization of that antigen specificity. So when we see a dark blue, that means there is a specific binding. If we see a light blue, that means there is no binding. We see that there are a large number of clonotypes that are specific to the influenza A peptide as well as the COVID spike peptide. We have moderate size clonotypes. We have a few clonotypes that are specific to the EBV peptides. And we also see some very small, perhaps rare clonotypes that are also specific to the COVID ORF, the influenza B peptide, etc. When we see a large number of clonotypes specific to the COVID spike, we say, okay, that's great, because we know that the patient has recently recovered from the COVID-19 disease, and we expect that. But for example, when we see that there is also a large number of clonotypes that are reacting to the influenza A, it might mean that the donor had a recent flu infection as well. It is possible, or maybe this is coming from a flu shot that they got. So these are some very interesting patterns that we can see. Another thing that we can see is that requires further investigation is it appears as though some clonotypes, for example, over here that I am pointing at the bottom of left-hand side, bottom of the left-hand side of this heat map, there are some clonotypes that are dual specific. They are reactive to both flu and COVID spike peptides. Is it because there is similarities there? this would be interesting biology to investigate. On the right-hand side table, there is a summary of how many clonotypes each of these peptides are binding to, how many cells there are, and one could imagine that for any of these peptides, doing a time course for a BMT experiment would also be very informative and interesting to study. Next, I want to show you the data from the BEAM-AB experiment, where we inquired what are the antigens, COVID antigens that the BCRs are binding to. Let me take you through this plot. So similarly, from the BEAM-AB experiment, when we have the gene expression VDJ and the BEAM readout for the feature barcode, what that allows us to do is gather the clonotype grouping data for the B cells for this donor. So that clonotype grouping data was taken, and nclone was used to build this plot called a honeycomb plot in nclone. In Loop browser, this is known as the clonotype distribution plot. Each dot in this plot represents a cell, and when the cells are grouped together, like you see 4 cells here, and then this large group in the middle, these represent clonotypes. The larger clonotypes are situated in the center of the plot, and then we have the smaller clonotypes at the periphery, which gives us this belt of single cell clonotypes over here. When we look at the plot, we have 3 different colors. So the green color represents the COVID spike, that is the original from the original Wuhan strain. We have the yellow dots, which are reactive to the D614G COVID spike. That's the mutation. And then for the purple cells represents cells that were specific to both of these spikes. And this table on the right-hand side summarizes how many clonotypes and cells were specific to each. That gives you a more granular view of the data. It was very interesting to see in this data that the largest clonotype that we identified was mostly specific to the original COVID spike. And there was also a fair number of dual specificity to both the original spike and the mutated spike. We don't see a huge clonotype for just the mutated COVID spike, and that might suggest that for this donor, their infection mostly came from the original Wuhan strain, and that's the strain that they were infected with. And here again, I forgot to mention that when the antigen specificity was calculated, it was comparing the UMI of the target spikes to the HSA antigen, to that of the HSA antigen. So that's how these specificities were calculated. Now that I have talked about the BEAM-T data and the BEAM-AB data, I want to talk about the large scale immune profiling data that was done on the 100K cells. So for these 100K cells, here is the UMAP. So let us first focus on figure A on the left-hand side. For those 100K cells, they were processed in multiple gem wells, obviously, and then those data were aggregated. From that gene expression data, we were able to carry out the usual QC steps, clustering steps, and dimensionality reduction steps to get to this UMAP that you see. So in this UMAP, there are all the population of cells that you would expect to see in a PBMC sample. But here I have highlighted the groups that we are most interested in. So here in yellow, we have the CD8 effector cells, we have the CD8 memory T cells here. These are in green are the CD4 memory T cells, and the purple are the CD4 naive cells. Over here in teal, this cluster at the right-hand corner of figure A are the B cells. Just because this is a bigger cluster with the blue, I'm going to just say that these are monocytes, and then over here in a tiny little bit, we have the platelets. So When we are doing this kind of a large-scale inquiry of the single cell, it is really helping us get a whole landscape understanding of what these donors' cell types are looking like. These cells, 100K cells, also have VDJ data. So for all of those, we also have full-length VDJ sequences and clonotype grouping information. What that allowed us to do is take those receptors that are antigen on peptide specific for BCRs and TCRs respectively, and map it back to this data. So that is what you see on figure B of this on the right-hand side. So at the top, if we look at the cluster that is corresponding to the B cell cluster on the left-hand side, you can see that the antigen specificities to the two different COVID spikes are all in this cluster, which suggests that we have been able to map those receptors back to this data. If we look at the bottom, if we come to the lower half of figure B, what we find is that if we map back those specific peptide-specific TCRs back, most of these receptors are now where we would expect them to be in this big place where the CD8 and CD4 memory cells are. Most of the colors that pop out of this spatter of colors is the blue for the COVID spike, and then the pink for the influenza A peptide. So that means that we are seeing a lot of these receptors are in the CD8 memory and CD4 memory T cell subtypes in this patient. But can we quantify? This is great. This is a proof of concept that we are being able to map the receptors back on these T cells. But then, can we quantify this? Can this give us more information about what kind of T cell subtypes are we seeing for each of these? So we chose a few peptides and we carried out that analysis that I'm going to show you on the next slide. So here, what we have done is we have looked at the flu peptide, the COVID spike, the three EBV ones, CMV, and COVID ORF1. And we have quantified from the previous, from just the previous slide that I was showing you, how many of these receptors are in naive T cells, which are in the dark bars, or in effector T cells in the yellow bars, or in the memory T cells, which are the red bars. For both flu and COVID peptides, what we find is that a larger proportion of these receptors are in the CD8 memory cells compared to the CD8 effector cells. Same with this EBV peptide. However, we do realize that an interesting pattern on this bar graph, which is that for the EBV BLF peptide, we see a little bit higher effector T cells compared to memory T cells. And we have wondered why this could be. While this warrants more deeper investigation, we hypothesize that it could be a sign of a latent infection. The donor has recently undergone stress in their immune system due to COVID infection. So maybe this infection is just recurring or coming back, which is why we see this kind of a pattern. But these are the kinds of things that the BEAM product will help us identify and also study. With this slide, I wrap up my discussion for this experiment, but I do want to draw some conclusions. I want to draw your attention that with a BEAM technology, we were able to identify BCRs that were specific to two variants of the SARS-CoV-2 spike protein. We were able to identify TCRs that are specific to multiple viral peptide MHCs. We were able to obtain full-length paired VDJ sequences for specific receptors. And also, we have shown with the data that with the BEAM technology, the development of therapeutic and diagnostic tools can be accelerated. And with that, I end my talk. And I also want to thank all the current and past members at 10x Genomics who has played a vital role in the generation and analysis of this data during a time when everything was shut down, and getting materials and getting everybody on the floor was challenging. Staying healthy was challenging, but they did it, and this is the crucial experiment that helped set forth the path for product development of Beam. So with that, I am going to end my talk, and I'm happy to take any questions.
Thank you very much, Nur-Taz. It was a very interesting, exciting technology. I'll let first the other people ask questions. I do have several, but first, let's see if someone wants to ask something.
Hi.
Oh.
Hi. Hi. Yeah. Thanks for the talk. You may have discussed this, but I was trying to understand. When you do the multiple viral peptides for the BT experiment, I mean, what is the constraint there? I guess number of cells per sample. You can't do too many viral peptides with, is there a technological constraint, or is it more that, well, you can't put too many in without kind of losing the specificity of your data, or just what is that? How would that work?
Thanks, Felix. That is a great question. So currently, we are able to, at the same time, inquire up to 15 peptides with, one peptide plus one negative control peptide, so 16 peptides in total. We highly recommend having the negative control peptide in there. You are right that the constraint is that if we put too many of the peptide of the tetramers in there, then it's difficult to understand whether the specificity we're seeing is because there is something specific there, or because a lot of non-binding, non-specific binding or switching is happening.
Okay, thanks.
Sure, thank you for the question.
So I see several people raise their hands. Let me just ask them one by one so that not everybody will speak at the same time. And I see also several questions in the chat that we can discuss afterwards. So maybe Encarnita, I think you raised your hand one of the first.
Thank you, Gur. Thank you, Nur, for the great presentation. So I have a question. How does that compare with the Immudex approach or tools for identification of antigen-specific T cells that initially were used and compatible with the 10x platform? And then I have another question. But
Okay, let me answer your first question. I had the same question as you when I first heard about this product, and I learned about the Beam T flavor of the product. The main advantage of the Beam T is going to be definitely in the pricing. My colleague Jose is here, who's a product manager for Beam. So Jose, please chime in. So it is going to be a much more easily accessible product because of the pricing, and also the software analysis and workflow for Beam T is completely supported by 10x. While we do not fully support the dextromers that you get from Immudex, you can definitely use it, but with the workflow, etc., there is not a full guidance that we can give our users for them because we don't have all the reagents under our control. It's somebody else's product, and we feel better giving you full support for the workflow as well as the software analysis.
You mean in terms of, for instance, the threshold that you put to determine whether a cell is specifically binding this tetramer compared to the negative control. This is what you are not supporting for Immudex because, or the dextramers, because they are not yours.
Yes, those are difficult for us to determine when it is a third-party product, so to speak. But for BMT, we are going to provide you with numbers that correspond to what the antigen specificity is, and you will be at liberty to set where you want to see the threshold. It depends on your donors, it depends on your peptides, you know, what is binding for you versus not. We will, with the BMT for 10x, and thank you for the question because I had forgotten to mention this, is there will be a negative PMHC with the conjugate already provided, so you don't have to worry about the negative control for the BMT at all. And that will be already provided, which will help our software calculate that kind of a score for you, and you determine where that threshold is for you.
Okay. So, and the second question was more about the result that you introduced, and especially the UMAP, where you are showing the different T cell subsets. I'm sorry, I'm more a T cell person, so focused on those ones.
Of course, yes.
So maybe I missed something, but you use class 1 tetramers, right, in this experiment exclusively?
Yes.
Yes.
A0201.
Yeah.
allele.
Okay.
Yes.
And still, there are few cells in your UMAP that seems to be CD4 cells that were binding those tetramers. So how do you handle them? Do you believe that the binding is correct and it's a real binding, or did you discarded them because of a threshold or whatever reason?
That is a very good question. Let me try to see if I can if I can quickly share again just the slide. I won't go into slideshow mode, but what I wanted to emphasize is these TCR overlap on this UMAP is coming from receptor sequences that we grabbed from our BMT experiment. So what that means is that we are finding those receptors in this CD4 population. So this is interesting biology that the CD8 and the CD4 probably have similar looking receptors that need further investigation, but these are not the exact receptors to which we have seen binding.
Okay.
Does that make sense?
Okay. So it's identification of same TCRs, alpha beta chains, but not necessarily T cells that bound the tetramer class 1. Okay.
Exactly. Yes.
Okay.
Yes.
Thanks.
Okay.
Of
Thank
course.
you. Before we go to the next two questions of Mark Lee and Matt Solin, maybe two questions, short ones from the chat. One is asking, Kieran Eller asking, how is it compares to LibraSec? And the other question is by KLG, which I don't know who it is, but if you confirm,
it's Ken.
Oh, okay. So if you confirm the TCR specificities with other approach.
I see. Okay. Let me first answer Kira's email. I am wondering if Jose, do you want to speak a little bit about how BMAB compares to LibraSeq?
Sure. Yep. Thanks, Nur. So, LibraSeq as a homebrew academic tool, you have the ability to, barcode antigens and then create these tetramer subdividing complexes, and then utilize, the 10x Chromium system to generate the data of interest. However, here you will have all of that workflow optimized for you, and especially what we have done is to optimize the staining, in which you can clearly separate out the antigen positive cells, and the control here would be something that is just the background complex, and all of that is integrated into the workflow with the fully kitted solution. So in a way, this is allowing you to not having to barcode your individual antigens. Here with the Beam app compared to LibraSeq, you come with just your biotinylated antigen rather than requiring you yourself to create individual
Jose, we lost your voice.
We cannot hear you.
Can you hear me now?
Yes.
Yes.
Oh, okay, sorry. The headphones are a little messy. So yeah, I mean that's the biggest difference from from this approach. And as Nur went over here, from the fully integrated software solution where we have optimized these interactive visualization capability, when the homebrew technology such as LibreSeq may not be fully incorporated into a software solution here as well. So overall, we just wanted to make it more easily accessible for all labs who can now, get started quickly with our kitted solution and go from there.
Thank you. What about the second question, Nur-Taz?
Yes, yeah, thanks, Jose. So for the second question, no, we did not systematically do validation of the TCR specificities. There were some ELISA spot experiments that were done to make sure that these peptides would bind to this donor before the experiment was done, just so because these were the early days, so we wanted to make sure that we were inquiring peptides that this donor would bind. And we did look through for the same peptides. We did extensive literature search to make sure that these are peptides that we have seen, that have other people have seen binding to in other samples, obviously. But to answer your question in a most straightforward way, no, we did not go back and confirm, but we live with this data and we have the samples too, and we hope to do more studies on them in the near future.
So, but just to add to Noor's point, for this question for the TCR specificity, we did not, take it to a functional study. However, what we didn't show here is that we, on the BCR side of things, we did have, the ability to clone the BCR clonotypes into an IgG, backbone, and did some binding specificity against the spike proteins, and have shown that the clonotypes that we are pulling out were showing good binding to the BCR, good binding to the COVID spike proteins itself, and also have done some follow-up live neutralization assay for the BCR side of things, which we haven't shown here, but we have tested the functional assay side of things to validate on the BCR side of things. As Nur pointed out, on the TCR side of things, we haven't, further confirmed from this data set as it is. Yeah.
Thank you. So let's go to the people who raised their hands. Maybe Mark Lee, can you?
Oh, yeah, thanks. So my question is related to Ken's question, which is that you showed that there are these T cells that appear to bind to both a flu peptide and a COVID peptide. Was there any sequence similarity in those peptides? I know you showed the sequences early on, but I couldn't recall. And then the follow-up question to that is there any possible way that could be an artifact? For example, like cell doublets, or I don't know. I'm just like thinking whether that you could think of anything like that.
That is a great question. Is this, is this real? And that's, I think, what requires more downstream investigation. In general, I think that this could be a little bit of, if it is not real, then it could be a little bit of maybe stickiness just in the receptors themselves, that there is some crosstalk, some similarities there. It is possible this could be non-specific binding in that sense, so to speak, a little bit as well. In terms of whether this is multiplets or doublets, working with VDJ for now, the VDJ algorithm now for a couple of years, there are lots of filters in place actually that prevent multiplets from getting into the clonotype grouping data. We try to control for the number of chains we are finding. We control for, UMIs that we are seeing so that we can get rid of those multiplets. So I would hazard a guess to say that these are likely not coming from doublets, but this could be non-specific binding. And it definitely looks interesting and warrants deeper investigation.
Thank you.
Thanks for the question, Mark.
Matt?
Yeah. Thank you for a great presentation. So when you looked at the antibody data, weren't you surprised that such a large fraction was specific for either one of these two very similar spike proteins? And how do you interpret that? Or were there other differences that are not captured, or that apart from the mutation at 614? Or if not, are there big structural differences between these proteins that translate into different antibody reactivities?
That is a great question. I do not know actually that if there are huge differences between these two spikes in terms of their structure. I do know that this D614G spike had an increased infectivity, like a higher rate of transmission compared to the original Wuhan strain. I think that there is still a little bit of similarity there, which is why we see a lot of dual specific VCRs here. But yes, it does strike us as surprising, but we haven't been able to follow up with that any further yet, but we hope to in the near future. Thanks, Max.
Okay, I see there is one more question that did not get answered in the chat, but before this, I will ask a couple of other questions. Did you look at the receptors that, well, the TCRs, did you look at their probability of generation? Are they common? Are they rare? I mean, are you familiar with Pigeon by tools like Sonia that tries to estimate the probability of generation of these receptors by chance? So did you look at it in this?
No, we have not done that analysis on this data set, no. But yes, that would be a very interesting one to do for sure. Thank you.
Yeah, which leads me to the, well, obvious question in iReceptor Plus seminar. I mean, do you plan to release at least a sample of this data that other people can look at it? And if so,
yes, we do. We absolutely do, and yeah. So Jose was mentioning that some of the downstream work that was done with coming from the Beam app data, and we hope to have that out published very, very soon. So yes, please stay tuned.
This is great. Another question is, did you look at the number of antigens per cell? I mean, since you have a barcode, right? And I guess you can quantify how many copies you have of the antigen in a specific cell. Did you look at it? I mean, do you have any kind of distribution of how many antigens are binding each cell, both for T cells and B cells?
Great question. So although I don't have that data, I don't have that information for this set of data, during product development and our internal studies, we are quantifying for that, and we do look at that. And when Beam is after Beam is shipped and it's available in the web summary that Cell Ranger provides, this information will be there, that how many in terms of how many UMI counts per cell are coming from the antigens. So that information will be available for our users of the product. But I don't have that information for this data. I apologize.
Okay, this is interesting. Another, well, last question for me is that I'm not sure in this type of data, but probably yes. I mean, if you just look at the receptors, you get many times that you have for the same cell heavy chain and then lambda and kappa. In these cases, how do you choose the right light chain? I mean, how do you know what is the productive chain of it? Do you have any thoughts about it?
So we trust the data that we get from the Cell Ranger VDJ analysis pipeline. And in that pipeline, we do see sometimes that there is a receptor with a clonotype with one heavy chain and two light chains. So in that case, we we trust it. We have some publications, some evidence to believe that there can be up to 4 different chains in a receptor. So we do not want to remove that with filters for our users. We want them to get the full data. But having said that, we have worked with users who like to look at only paired sequences with 1 heavy chain and 1 light chain combinations. In terms of whether, if you see 2 light chains, which one is productive? So the ones that the assembled contigs that VDJ filters out are always the ones that are productive, and there are 5 different criteria in the algorithm for what is productive. One thing I know that our productivity criteria is a little bit more stringent than what's there in the IMGT request. But if we see, so my point of saying that is, if we do see 2 light chains and a heavy chain in a clonotype in a receptor, we tend to believe that.
Thank you. So I think there's a last question by Savita. Maybe he or she would like to say it out loud.
No, I just thanks, good. Actually, I just remembered this paper that I had seen where SARS-CoV-2 infection triggers other viruses to be activated, which was interesting because what Nur showed kind of validates that.
That is fantastic to know. Thank you so much for sharing the publication.
Thank you so much for the lovely talk. Thanks.
Thank you very much. I think there is a last comment by Brian, who says that there is a new single cell repository for the iReceptor, and you can collaborate and host your data there.
So I was about to type a comment for there, particularly in this COVID study, there's a much more larger study that we're collaborating with some group from academia who basically will be the ones who's driving that. So we may not be able to really, tell you if that is shareable. Depends upon the academic collaborators what that will be. But some of our internal data sets that we will generate, is something that we would be having full control over in terms of how we can share those studies with you guys.
Yeah, that would be great. And if you want to mention this capability to your academic collaborators, and if they're interested in sharing, it'd be great if they could reach out to us.
That sounds great. I'll follow up to get your contact information as well. Yeah.
Great, thanks. I'll put it in the chat.
Okay, thank you so much, Brian. Yeah.
Okay, so thank you very much. It was extremely interesting. Let's take 5 minutes break and come back at the round hour for our next talk. And if someone has more questions, they can put it in the chat and people can react.
Thank you.
Back at the round hour. And thank you very much, Nur-Taz and Jose.
Thank you. Take care.